Chapter 19 is all about lipids. It's a little bit of a shock to go from carbohydrates to lipids because while a huge number of monosaccharides were really just stereoisomers of each other, not even having different things connected, just different orientations, lipids have no universal structural feature. Lipids are just generally defined as a biomolecule that's not very soluble in water, but soluble in nonpolar solvents instead. Again, lipids don't have anything that's universally true for all of their structures. They do have a tendency, though, to have lots of carbons that are single bound. We have two major categories, saponifiable lipids and non-saponifiable lipids. A saponifiable lipid is essentially a lipid that we can hydrolyze into a soap. And all of these will contain either esters or amides. If you look at the examples shown on the screen, every example with a portion highlighted in green has either an ester or an amide. The green piece is the carboxylic acid portion of these esters or amides. These are all saponifiable lipids. The only example shown for a non-saponifiable lipid is the steroid that's on the top right. Most non-saponifiable lipids tend to be based on the same four-ring steroid structure and do not have esters or amides. In this chapter, we'll divide up lipids based on roles. All the lipids in red are saponifiable lipids, while those in blue are non-saponifiable. In general, lipids are structurally similar when they play a similar role, with cholesterol being an exception. All saponifiable lipids share the same form for the carboxylic acid part of the ester, or amide. We call them fatty acids. So for this first lesson, we'll talk about fatty acids and their properties, much of which repeats from chapter 13 and chapter 16. We'll also talk about the main way that these fatty acids are stored for energy, and therefore the main way they enter our diet and metabolism, energy storage lipids. In the next lesson, we'll tackle all the other roles of lipids. Fatty acids are, as the name implies, acidic. We know exactly one significantly acidic functional group in this class, the carboxylic acid. Turns out fatty acids are really just long carboxylic acids. The simplest fatty acids are saturated fatty acids. That would be fatty acids that only have single bonds between the carbons. This corresponds directly to saturated fats in a nutrition label. Saturated fats are fats whose fatty acids are all carbon-carbon single bonds. Conversely, unsaturated fatty acids have one or more double bonds. Your body can make as many saturated fats as it needs, but it has trouble making unsaturation at certain locations. So generally, unsaturated fats are more important in your diet. Monounsaturated fats have just one double bond. Polyunsaturated fats have more than one double bond. They're generally more healthy and are more common in plant-based food and cold water fish. No matter how many double bonds are in the fatty acid, though, the naturally occurring bonds are always cis. We never have trans fatty acids dietarily. Most fatty acids have trivial common names, often related to the original source, meaning that each one would need to be memorized individually. We're not going to do that. There's also a shorthand that can identify the fatty acid by length, the number of bonds, double bonds, and the positions of those double bonds, starting at the carboxylic acid carbon. The first number in this form that's given is just going to be the number of carbons. In our examples, in all of our examples, and most examples in general, this will be an even number. It's fairly common for people to either count an oxygen from the carboxylic acid as a carbon, or forget to count the carbon on the carboxylic acid. In either one of those cases, the number would show up as an odd number. So this can be an easy way to check yourself. You shouldn't get an odd number. After the number of carbons, we're going to put a colon, and then the number of double bonds. If that number is 0, we don't need to add anything else to this molecule. But if there are double bonds, we put the Greek letter delta, D delta for double bonds, in parentheses with each of the double bond positions as superscripts, separated by commas. So the fatty acid above, you can see the name of in the bottom right of this paragraph, 18:3, parentheses, delta 9, 12, 15, and then a close parentheses. Try to name the following fatty acids using the same shorthand. The first fatty acid has 20 carbons and 4 double bonds, so 20:4. The double bonds are at positions 5, 8, 11, and 14. They should always be spaced 3 apart. If you got a different number than 5 for the first of the double bonds, make sure that the first carbon is the COOH carbon. The second molecule is 20, 5, delta 5, 8, 11, 14, 17. And the final fatty acid is 22:6, delta 4, 7, 10, 13, 16, 19. In addition to this shorthand, we also categorize fatty acids based on the position of their last double bond. Carboxylic acid carbons are informally given Greek letters to refer how far away from the carboxylic acid they are. Alpha would be the first carbon off the carboxylic acid, beta the second, and so on. So the very last carbon is the omega carbon. The lowercase omega looks a lot like a W. So if we count 3 carbons from the end without a carboxylic acid, the omega end, before we see a carbon with a double bond, we'd call it an omega-3 fatty acid, like the example on the top. See if you can categorize the bottom 3 fatty acids shown in this way. In the first example, the omega carbon is on the left side. Counting from the left, we have 6 carbons to the double bond, making this an omega-6 fatty acid. The second molecule is an omega-3 fatty acid, as is the final molecule. Only 2 fatty acids are necessary parts of our diet. Anything that we cannot make in our bodies and have to get in our diet, we call essential. Everything else is non-essential because it can either be synthesized from these 2 fatty acids or indirectly from carbohydrate sources. Notice that each of these fatty acids is unsaturated. Based on this, do you think saturated or unsaturated fats are a more important part of our diets? Because we can only get unsaturated fats from these unsaturated fats, but we can get saturated fats from anything. Unsaturated fats are an essential part of our diet. You should know the names of these 2 fatty acids, linoleic acid and linolenic acid. We've already talked about the physical properties of both carboxylic acids and alkenes. With very long molecules like fatty acids, we shift from talking about boiling points, which would be really high for these molecules, to talking about the melting point, which is closer to the temperatures we can look at in the lab. But the trend remains the same. The line at the top of the graph shows the melting points of saturated fatty acids going from 12 to 24 carbons. Notice that we increase by 2 each time. Even-numbered fatty acids are by far the most common and the most relevant biologically. As the number of carbons increases, the melting point does too. Just like any other time we looked at a molecule type, the boiling point or melting point always goes up with more carbons because the number of London interactions increases as well. Since all the saturated fatty acids melt above room temperature, saturated fatty acids tend to be solid. When these fatty acids are part of a dietary lipid, we call these solid dietary lipids fats. Fats tend to be from warm-blooded animal sources, which can access these in a more fluid form because they're heated, they're warm-blooded. On the other hand, if we look at a straight line down from stearic acid, the straight line in the kind of rightmost part of the graph up from 18, we can see how the presence of one or more double bonds in the molecules changes that melting point. You can see each of the structures shown on the right. Even one single bond drastically reduces the melting point, as the permanent bend that's introduced by the cis bond reduces contact between the fatty acids and causes them to interact more weakly. Unsaturated fats then tend to be more liquid, and they are often sourced from either plants or cold water fish. We typically call liquid dietary lipids oils, hence animal fat, solid, and vegetable oils, liquid. One of the most interesting dietary discoveries with regards to fats is that even though some people groups, like the Inuit people, traditionally consume diets with far more fat than the typical diet in the US, the Inuit diets aren't associated with the same types of diseases that are associated with high-fat diets here. The major difference is the amount of omega-3 fatty acids in the diets. The diets of Inuit persons tend to be very high in cold water fish, which in turn are relatively high in omega-3 fatty acids. While plant-based oils also tend to be unsaturated, they don't have as high of a ratio of omega-3 fatty acids to omega-6 fatty acids. And animal-based fats in general, besides cold water fish, tend to be more saturated. Fatty acids alone don't make lipids, and even the lipids that contain fatty acids do so as part of esters. This controls the impact of the acidity of fatty acids on your body and also allows fatty acids to be stored much more compactly. Remember that the major way that we store carbohydrate energy is glycogen. Glycogen has tons of alcohols, which makes it fairly polar, and it can be stored in most tissue types. Energy storage lipids are a much more efficient way to store energy, but because they're mostly nonpolar, we need to store them in special cells, called adipose cells, which are in turn part of adipose tissue. Energy storage lipids are all one type of molecule, triacylglycerols. The acyl part of that name just refers to a carbonyl group that's attached to something electronegative. When we've looked at reactions before, we called this the carboxylic acid portion of the molecule. So the triacyl really just means that there are 3 carboxylic acids that are part of each of these molecules. Hopefully you can identify that those carboxylic acids are probably fatty acids. The glycerol part refers to the specific molecule glycerol, or 1,2,3-propane triol. If you didn't memorize it back in chapter 14, memorizing the glycerol structure now is pretty important. It's just 3 carbons with an alcohol on each. Triacylglycerols are esters between the fatty acids and glycerol's 3 alcohols. You should know this structure in a generalized form, a glycerol with 3 fatty acids on it. Triacylglycerols are not just one molecule, they're an entire class of molecules. You could have any combination of any fatty acid in any order. So triacylglycerols may consist of many saturated fatty acids and be solid at room temperature, a fat. Or they may have mainly unsaturated fatty acids and be liquids at room temperature, an oil. We have a couple of different types of chemical reactions that can happen to triacylglycerols based on the functional groups that are present. We see in a triacylglycerol the main functional groups are esters, and we know that esters can hydrolyze, break down into the pieces, the fatty acid and the alcohol, or they can saponify, which is the same thing but in base. So we'll actually see this happening, and that's how we make soap. We also see it happening in our body to release fatty acids for energy. We also have alkenes and any unsaturated fatty acids of the triacylglycerols. Those alkenes can be converted to alkanes by hydrogenation. It's one of the reactions we talked about in chapter 13. These triacylglycerols usually don't fully react in any of these cases. They're big molecules and they're hard to access the inside of. And so often we have partial reactions. So we might have partial hydrolysis, which reacts maybe 1 or 2 fatty acids, but not all of them. Or partial hydrogenation, where some of the alkenes are converted to alkanes, but not all of them. Partial hydrogenation always has a byproduct, trans fat. Whenever we start to convert an alkene to the alkane, it can be reversed, and when it's reversed, it always forms a trans fat, which is more stable. And even if we try to fully hydrogenate, there will always be some amount of trans fat that's present. And so whenever we look at a nutrition label, if we see hydrogenated any sort of fatty acid or energy storage lipid, we know that we will also have trans fat.